Hello and welcome back. So we have a session with Dr. Naresh Selokar. We'll be talking about buffalo cloning technology for conservation of endangered bovine species. He is associated with Animal Biotechnology Center, ICAR NDRI Colonel. I would invite Dr. Sambasiva, scientist CCMB, to introduce our speaker for the session.
Thank you, Amir. I'm happy to introduce Dr. Naresh Selokar to the participants. Dr. Naresh Selokar is the scientist at the ICR National Dairy Research Institute, Karnal, India. He studied veterinary sciences at the Nagpur Veterinary College and earned his master's and PhD degree in animal biotechnology from the ICR National Dairy Research Institute, Karnal. He completed his 6 months of the Commonwealth Fellowship training in cattle embryology at the Ontario Veterinary College, Canada, and 1 year postdoctoral training on CRISPR-based genome editing in mice embryos at the FLI Institute of Farm Animal Genetics in Germany. Dr. Selokar authored more than 40 scientific publications in peer-reviewed journals. He has received several awards, including most recently the Associate Fellowship, NASA India, Young Scientist Platinum Jubilee Awards for 2019, and Innovative Young Biotechnologist Award for 2018. The Young Scientist Award 2015 is CIRB India, and Jawaharlal Nehru Award for Outstanding Doctoral Thesis Research in Agricultural and Allied Sciences. Now I invite Dr. Selokar to share your presentation and go ahead.
Hello, I am audible, sir?
Yes, you're audible. I would like to remind you, Dr. Naresh, that we would be having 35 minutes for talk and 10 minutes for question answer. Thanks a lot.
Okay, thank you. Thank you. First of all, I'm really grateful and thankful to Dr. Umapati for his invitation to deliver the talk is this ADVETCON Conference 2021. And also thankful to Dr. Sambasiva for the nice introduction of mine. The topic. Yeah, slides are visible?
Yes.
Yeah.
Hello?
Yes,
it's visible.
Okay. The topic I choose for this presentation is the buffalo cloning for conservation of endangered bovine species. Though my majority of work, I work in the domestic animal, which includes the majority in buffalo. And now I have been at NDRI work initially on the cattle too. But some of these previous work we did on these other bovine species, for example, the wild buffalo or the Assamese buffalo, and try to clone them through the somatic cell nuclear transfer technique. So the cloning, I think everybody those are working in the reproductive biotechnology, they are much aware about the cloning, how it's similar to the dolly as well trained sheep in domestic animals. So actually the cloning is the making of biological copies of any individual. It's a sexual mean of reproduction, in which there is no need of the spermatozoa to make an embryo. So the somatic cell from any animal, either it is a wild animal or it's a domestic animal or it's a laboratory animal, that differentiated somatic cell transplanted into the enucleated oocyte, the genetic material of oocyte to be removed, and then after the embryos to be activated and never to be blasted stage and then transfer and produce the clone animal. That is the overall method. This is just a pictorial presentation of how we did the buffalo cloning in our institute. So we took the cells, we brought the ovaries from because in buffalo we have the ample amount of the ovaries to get an oocyte for these all the assisted reproduction. The method we used in our lab is the handmade cloning, which we see in this picture is that the two enucleated oocyte is a sandwich between two enucleated oocyte, one somatic cell is sandwich. So this is a method we did in India. So and through this method, at least in buffalo, we can say at present we have the 21 clone animals in the two institutions. Previously, I worked at the CIRB, Central Institute of Research on Buffalo at CISR. There we have the 11 clones, particularly elite bulls, and at the NDRI we have around these 10 clone bulls, and we are continuing to produce more clones, particularly for the buffalo breeding bull. Now, this is an overall journey picture of why I would like to mention is that the cloning, the technique we developed in the buffalo, because in principle, if we have the technique in hand, So this technique can be extrapolated particularly for the conservation of endangered or these endangered, not endangered domestic animal as well as for the wild animals. So there are some domestic animals, for example, as the Pangunur cattle from the Andhra Pradesh. They are being critically endangered, and the government is planning to conserve them through the either assisted reproduction, semen cryopreservation, somatical banking, or OPU, IVF. Similarly, the Ongole cattle breed. And similarly, for example, the wild buffalo, which has been previously the lone buffalo in Chhattisgarh Wildlife Sanctuary, or other domestic animals, these gore or these the wild oxes or other things can be conserved if we have the technique in our hand. So, and if you go through this technique, the buffalo cloning in India in 2009, the first clone buffalo is produced in the country by the National Dairy Research Institute, where I did my PhD. At that time, I was doing my PhD at NDRR. At whatever these are overall pictorial presentation, what we have currently with our hand. So we got the embryo production rate is ranging from 30 to 35%. So live birth rate is a 6-8%, which is later than that of conventional artificial insemination or the embryo transfer technique. But we have improved this technique from 2% to at least we have the 6-8% live birth rate. The other drawback we can say in this technique is that we have a huge number of pregnancy losses. So usually what we do at the day 30, we check the animals through the ultrasonography, these animals are pregnant or not. But when we check again at a monthly interval, we did at least for 4 or 5 months, and we noticed that once those animals, those are pregnant at the 30 days, the 20 to 30% of animals they are not pregnant at the 60 days. So there are huge embryonic losses at the early pregnancy losses. We have around the 25 to 30% pregnancy losses. And during our optimization, the different type of somatic cells we use on the skin-derived fibroblast cells, stem cells, urine, and semen-derived somatic cells. Almost whatever the cells we wanted to explore, so we tried which somatic cell works better for the reprogramming. And at the present, for all the cloning experiment, we use the skin-derived fibroblasts derived from the cell, base of the cell. So as I mentioned, as of 2021, we have 20 clone buffaloes. These buffaloes are alive, more than the 35 or 40 buffaloes we produced through this cloning, but some of them, they have been died in early age or because of the abnormalities, but still we have the 21 clone buffaloes, of which the 16 are the breeding bulls. And recently, my previous organization produced the 7 copies from the single bull, as well as attempted and produced the recloned calf from the earlier clone bull at the Hisar. So recently we also studied and shows that those are the clones, they have maturity, either is a male and female, they have the normal growth, and they have the good health. We checked these his serum biochemistry or hematology. Also, we did the artificial insemination from the clone bull semen, as well as for the natural artificial insemination of the clone females, and the progeny is produced. At the NDRI, we have some females that has delivered the 6 calvings. So as well as the bulls, from the bulls, we have around produced the 25 progenies, 20 to 30 progenies from the 1 bull from the CSR and 2 bulls from the NDRI. And the progenies of the bulls, the clone bulls, they are absolutely normal. As well as we check production, for example, in case of the males, we determine the semen production parameters, for example, the CASA indices, as well as in case of the male, the milk composition, their constituents, they are absolutely normal. But we are talking about those are bulla, either is a male or female, they are mature well. So these are the overall pictorial presentation of the buffalo cloning in the country. Now, these are the 2 institutions. They are basically working on the buffalo cloning. One is my previous institution where I started my first job at the CIRB in 2013. And this year I moved to the NDRI to again the strengthening of the assisted reproduction. So these 2, because the NDRI has been pioneering in the field of the assisted reproduction, and they wanted to continue, and the majority of the, either it is an OPU, IVF, or cloning or transgenetic, so we have been taking up at the NDRI. These are some of the achievements, just the multiple production of the clone cows. Now coming into the major aspects. So these are, and I'm not going to detail what we have been done in the buffalo cloning, which is in intra-species cloning or same-species cloning, but I would like to just highlight the other experience about, for example, the cloning of the cattle or cloning of the horses and cloning of the Assamese buffalo and cloning of the wild buffalo. So we attempted out that out. We get some success in some of the species, some we could fail. For example, we fail in the horse cloning, and we miserably fail in the cattle cloning to the buffalo side. So normally there is, particularly for conservation of the endangered bovines, for example, either it's domestic animals or it is a wild animal, the major problem is the lack of the oocyte. And to mitigate that, we need to identify their contemporary oocyte from the domestic animal. And if we talk about in country, we have these buffalo slaughter is available in every state, and that's why we choose the buffalo as a model to study the reprogramming ability from the other domestic animal as well as wild animal somatic cells. If we have cattle oocyte, for example, in the Kerala or in the West Bengal, then there is a possibility that this oocyte from the cattle can be used to study that can be cattle oocyte to the reprogram of the differentiated somatic cells from the endangered species of the dairy animals as well as from the wild animals. What exactly, and this entire process is for the interspecies cloning, in which what we did, we need to use the somatic cells from the endangered animal and the enucleated oocyte from the domestic animal counterpart and study these and produce the embryos and then check the in vitro development if they have succeeded to reach to the blastocyst and what will be the quality. And if we have checked the quality, then to determine the in vivo developmental rate to produce the clone progeny. These are the first paper we conducted at the NDRI, in which to identify the ability of the buffalo somatic cells, buffalo oocyte, the enucleated oocyte to reprogramming ability to other species, for example, the cattle, goat, and rat. But in case of the cattle and the goat, we could able to produce the embryos, but that embryo production rate is extremely less. For example, 3% or 2%, not more than 5%. So embryo production rate is severely compromised. So we also, in case of the rat, if you use the rat somatic cells, then there is absolutely no embryo development. So the embryos will be blocked at the 8 to 16 cell stage. But the buffalo, they work well. So this indicated, though the embryo production rate in case of the cattle and goat is poor, but it's indicated that buffalo oocyte have some competence to reprogram the differentiated somatic cell of other species. For example, here is the case the cattle and goat. And this study, we continue to explore for these production of the wild buffalo. I think Dr. Umapati and Dr. Sambashiva here, they are well aware that the project has been assigned to NDRI. That time I was doing my PhD, then subsequently when the calf is born, I was joined at the CIRB. So what they did in these NDRI, they have the collaboration with the Chhattisgarh Wildlife Department and WTI also, and that they have wanted to clone these wild buffalo available at the wildlife of Chhattisgarh, available at the Chhattisgarh. That is for the name of the buffalo is Asha. So these are the comparative aspects of these the embryo development rate of the wild buffalo as well as the domestic animal buffalo. If you look at the blastocyst production rate, there is absolutely no blastocyst production rate are the same. There is no difference because they both are these buffalo species. So our study in 2011 study indicated that when the species are close, their embryo development is proper, appropriate, and they will be able to produce the normal embryo development. But when the species are taxonomically much deeper, then it's very difficult to produce an embryo. And to produce the live animal, the important thing is that the recipient oocyte has the potential to differentiate or potential to reprogram the somatic cells of their contemporary animal. If they're close-related, then easy to reprogram into the blastocyst stage. If they're far away taxonomically, then it's really difficult. But in this study, the embryo rate is absolutely normal, though there are a little bit cell number, total cell number is the less, half than the appropriate index is high. But these embryos can also be vitrified to see how these embryos of the wild buffalo evolve behavior well. And the NDRI has been claimed to produce the wild buffalo, and then subsequently it's handed over to the Chhattisgarh department. This is another study. What happens whenever we culture these cells in vitro from the tissues to be from, for example, biopsy? During this case, there are the two types of cells are established. Either is the epithelial cell or other is the fibroblast cell. So we thought, can it possible for us to determine the developmental competence of these both, either it's the fibroblast cells or epithelial cell, which cell is better? Because when we culture from these precious samples from the wild animal, then every cell needs to be studied. So we thought in vitro culture cells, there are two types, fibroblast and epithelial cell. We produced an embryos and we found that the epithelial cells from the same species, for example, wild buffalo, the same use, they are poorly programmed as compared to the fibroblast cells, because epithelial cells, they have a different expression pattern of the gene, and that could be the effect on their developmental competence. So if you see here, the blastocyst rate in case of the fibroblast cell is 50%, but in case of this cell, just an 20%, which is in half of that of the normally fibroblast cell. Though there is no difference in terms of the total cell number as well as the apoptotic index, but the embryo development is severely compromised. This is again we thought to go in the detail about which cell type is work well, although we found that the embryo production rate is compromised. But what are there in the molecular level? For example, histone markers. So different histone marker studies, H3 K18 acetylation, H3 K18 methylation, as well as expression of these epigenetic gene, apoptotic gene, as well as the developmental gene. So it found that those are histone markers. For example, those are suppression of the gene is higher in case of the embryos produced by the epithelial-derived cells, and these similarly that has been reported in case of the somatic cells. So the pattern is not changing. That there is a similar pattern they are expressing from the somatic cells to the donor in embryos. Similarly, when we compare in case of the clone embryos, if you see here some of the gene expression, for example, the NANOG. So NANOG expression in case of these epithelial-derived blastocysts is down-regulated as compared to the fibroblast-derived embryos. So the NANOG, OCT4, and then these are the two genes related to the pluripotency. Then other gene, for example, the CDX2 expression in case of the epithelial cell, they are high, but these are the genes responsible for the surfactant differentiation. Similarly, the DNMT and HDSC, these are related to the epigenetics. DNMT creates related to the de novo methylation pattern. If there's more, then there's more methylation, which has been also been expected from our immunofluorescence staining. So overall, from this study, we conclude that the epithelial cells are poorly programmed as compared to the fibroblast cells. Now, and this was the story from the wild buffalo we attempted at the NDRI, and these are the study we attempted at the CIRB during so we thought to clone horses because there are some demand from the horses. And in the Hisar, there are the military station, which has the name of produce the breeding horses for the military. So we thought this is in collaboration with the NRC Equine. This is another ICR institution is here. So we thought, can it possible possible that the buffalo side to be explored for the horse and produce horse embryos. So the horse somatic cells established. And then similarly, with the other interspecies cloning, this horse somatic cell is sandwiched between the two nucleotides and other parameters we studied, for example, cells, because the first time we worked on the horse somatic cells, so we studied the cell in vitro, cell growth pattern in vitro, the proliferation, the mean time, and the culture condition, what is required for the horse, and what are the parameters for the electrofusion parameters required for the horse somatic cells that we also studied. And then after the production of embryos fusion, we studied these embryos to be reached at the blastocyst level or not. If they reach the blastocyst level, what are the extent of their apoptosis level and then some of the gene expression patterns. So these are just a pictorial presentation. The samples have been collected from the Bikaner breeding station. My collaborator, Dr. Taluri Rao, so he was working as a gynecologist at the Bikaner breeding station from the NRC Equine. So he was also collaborated for this project. So we collect the samples from the different age groups, the newborn foal, the young filly, and the adult colt. So these are the, in the downside, you can see the pictures of the horse somatic cells. Once we have the cells, then we sandwich between the cells. In the first attempt, because with the conventionally we produce the two enucleated oocytes and the one somatic cell. So when we produce the embryos by the fusing of the horse, these embryos, they are arrested at the 16 cell stage. We could not able to produce the blastocyst stage. The second attempt, we thought, because maybe the horse, the embryos development is slow, they require more cytoplasm. So we sandwich the three enucleated oocytes and the four eggs. You can see the pictures in the left hand side. There is a doublet one, which is standard method we use for the buffalo, and the triplet and quadruplet we also tried to produce some of the embryos, but in any of the group, we could not able to produce the blastocyst stage, and we fail in this experiment that our embryos, they are arrested at the 62 stage is the maximum. So other approach, we thought to use the cells from the different age groups. For example, the adult horse cells, or the fowl cells, the newborn, as well as cumulus cell. But even if you use the different cell types, they will not be able to develop into the blastocyst state, which indicated that if as our previous study in 2011, if the species is severely diverse in taxonomically, they're difficult to reprogram, but that was, we need to attempt it. And from this study, we can say is that the buffalo oocytes cannot reprogram to the horse somatic cell. So these are the reasons why these horse embryos is not, maybe the embryonic genome activation is improper, or is mitochondrial defect, or suboptimal culture conditions. So we need to conduct more experiments to identify what problem in that. Either is a genome activation problem or mitochondrial defective problem, because the horses and the buffalo, they have the different mitochondria, how the compatibility issues, or maybe the suboptimal culture condition, because we have not attempted much experiment. Because just in pilot project we wanted to initiate. If we get in 1 or 2 blastocysts, then there will be the more hope for us to explore and improve the embryo production rate. So this needs to be studied. Then we thought to get an ovaries from the euthanized horses and the military reform CIRB. So somehow we have made an agreement with this army breeding station. Whenever they euthanize the horses, we collect the ovaries. But the problem is that, so again, because of the first time we are working with the horses, media or to extraction of oocyte is extremely new for us, and we struggle a lot. But in that, we got some oocyte. These are the pictures of the horses horse oocyte, but the majority of them, because we don't have the optimized media for in vitro maturation, we conduct the optimized medium, and we found in that the amount of hormones required for the horses, though we refer the previous papers, but that will not result it into the maturation. So some of our experiments failed because the oocyte is not mature at all. And then we found that the amount of hormones required, particularly for the horse oocyte, is almost 10 times than that of the buffalo, what we have the experience in buffalo. So other drawback, because even though we got the few oocyte, for example, 10, 15, but this number of the oocyte is not sufficient for us to conduct the SCNT experiment. So we did not do any of the SCNT experiment. We just tried the in vitro maturation, and then again is problematic for us to collect the sample from the euthanized horses. This is I think successful attempt we could made in case of the Assamese buffalo. So in Assam, these are the major buffalo. They are swamp type there, and these buffaloes they are mixing either swamp type or riverine type, and they're free ranging and then well adopted. Some of the people, they rear the buffaloes too. So we thought, can it possible for us to clone a buffalo of the Assam? So we thought to collect the first of all the biopsy from the Assamese buffalo. So we airlifted the sample from 3 Assamese buffalo to the CIRB, and then established the somatic cells from all the 3 animals. And then after that, the embryos, they are produced to our normal method of the cloning with the double cytoplasm, 2 cytoplasm and 1 enucleated cell. And in case of Assamese buffalo, we got a good number of embryos. If you see here, our embryo production rate is around 15 to 20%. Here, these are old data, so it's 15%. But with this time, we get the sometimes some animals use 30% embryo rate too. So we got the 15 to 20% embryo production rate, and there's a good number of embryos we can produce from that. Then these embryos we have transferred in our farm. Also we transfer these embryos into the field. So field means that there is one district CIRB, which is an organized dairy farm there. So this high-tech dairy farm, we transfer 6 blastocysts into the 6 recipient animal, 1 blastocyst per animal. Out of the 6, 1 found pregnant and that deliver the calf. So this is an Assamese stone buffalo born at the CIRB. So this was, I think, the first attempt we tried to produce at the field level, that as an embryos produced at the laboratory and then transfer into the breeding farm. And this is the organized breeding farm. So also previously, whatever the clones we produced, they were all are the Murrah type. So this is again, we attempted that other breeds of the or other species can, for example, the buffalo, even though this buffalo can be cloned. So we also attempted the Niliravi buffalo cloning, but we failed there. We got the 5 pregnancies from the Niliravi, but none of them is delivered the calf, and one calf has delivered, but that calf has been died in the 6-month period. So we don't know which species is work better, because in most of the time the oocyte we got from the Delhi slaughterhouses, majority of time they're Murrah type. So maybe is possibility that the other breed somatic cell is not appropriately reached to RAM during the oocyte. That needs to study. And in this study, we proved that yes, this Assamese buffalo, at least we can say, those are in the late in number, and the government is planning to conserve that. So it works well for the particularly for the recipient oocyte from the Murrah type. Also, is that in this case we use the surrogate animal, Murrah animal. If you see the farmers which is holding these Assamese buffalo clone bull with the surrogate animal, which is a Murrah type, black in color, you see exactly the difference of the in terms of their morphology or physical appearance. They're absolutely, it looks like a wild buffalo animal. It's exactly the downside. This is a donor bull available at the Guwahati Veterinary College. They have a captive breeding program. They run and they're herd, but I don't know. They are continuing with the samples has been collected in 2014, and I remember subsequently that center has been discontinued the breeding of these Assamese buffalo. Also, we use the single method of the embryo production. This is one research aspect. So through this entire study, it proves that the distant animal, for example, the Guwahati animal or from the Kerala or other animals, can be cloned by airlifting the sample, because sampling airlifting particular somatic is easy, and these embryos when produced that can be transferred to any breeding herd. This is bull as and now. So this bull is mature well, and then he's donating the semen at the CIRB. At present, his weight is more than the 450 kg, 487. And the birth weight is also higher in case of this animal. Usually, we got the birth weight around the 34 kg, but in this bull, Assamese buffalo, is the weight is of 54 kg, born in 2017. So we did the parentage verification. Obviously, it looks different, so there is absolutely clear cut that this is a bull from the Assam. We clone at our institute produce embryos and did transfer at the field. We also checked the chromosomal abnormalities and the serum biochemistries. This bull, they don't have any abnormality. And we initially thought that most of the animals in Assam, they are the swamp buffalo style, which is 48 chromosome number. But unfortunately, in our case, this animal has a 50 chromosome number. So it might be because there are the crossbreeding over the period of time, and this is bull from the breeding herd. That's why it's a chromosome number, or they have the mix, and subsequently it is a riverine type of buffalo, not and after there's a wild that is a span buffalo. So this is we also get it published in these cover in these Rajya Sabha TV as well as the current science. And at the present, this bull is donating the semen. We train this bull to produce the semen at the CIRB, and we have around 5,000 semen doses. We also check the CASA indices of this bull. So they're absolutely normal. We did the ideal breast fertility, and the current blast, we get the blastocyst around 23% of the blastocyst rate. And recently, we wanted to use the semen in the breeding tract of in Assam. So we approached to the Agriculture University Assam and Guwahati Veterinary College, or the ICAR institution available in the northeastern state. So at least to do some field trial, particularly for artificial insemination, to show that this bull can able to produce the progenies to the artificial insemination. We in vitro fertility we already checked. We found that in CASA there is absolutely no deviation in the normal indices, which we, for example, there is a motility indices or the linearity, the whatever the indices, it is the acrosome integrity, everything is normal. and the fertility, IVF fertility is normal. Now is the time to use this bull for the breeding at the northeastern state. What these studies from this Assamese buffalo, we can say the distance animal can be cloned. For example, the wild buffalo we attempted, but more attempt to be made to produce more number of progeny. It is possible to transfer away from the laboratory, because from our laboratory at Hisar and the field is approximately 100 kilometers away, and the embryos just transfer simply in the thermocol box with the ice pack. Ice pack means a warm ice pack. So there is no absolute incubator, nothing has been is there. So it is possible. Also possible to this study is it proves that the field animal can be used as a surrogate animal, because what happens sometimes in the institution, for example, NDRS, CIRB, though we have the animals, domestic animals, we have 500 animals, but in there, a lot of projects are ongoing. So we are scarcity of the recipient animal. In that situation, we can approach to the breeding herd or other organized dairy farms which have the 20-25 animals and request them, can we wanted to transfer some of the embryos. If you get an animal, then it's a possibility that can be done to the study. Also is that the distant animal north and south and south and north, if the cloning can be done. So the important thing is that what I would like to mention here is that this study has encouraged us to use the somatic cells from the wild bovine. And I'm eager to explore, for example, the Mithun domestic animal. I approached them to provide the samples from the Mithun, or even from the wild buffalo. In Maharashtra also, so for example, Gadchiroli district, it is near to my district. So we have these animals there, wild buffalo. So if they provide us wild buffalo, but again, the getting of sample is not so easy. So for example, so we are trying, so I'm trying to get samples from some of these wild animals, for example, the boar or the bison, to just explore how it works well. To either we will get the embryos. If we get the embryos, then it's good to get and hope to produce an animal. If we have not getting embryos arrested, still we have generated the information and knowledge that this species not possible to clone through the buffalo side. The another study, this is all about the buffalo. Another aspect, I think, at CCMB, Dr. Samba Sivaya, as I know, he's taking care of the establishment of somatic cell biobank. And this is again a good approach, which I believe also is very important to establish the somatic cell biobank of this wild animal as well as domestic animal too. Because if your wild animal is good enough, because in future you don't know, suppose somebody wanted to use the somatic cells for the generation of induced pluripotent stem cells or for generation of the clone embryos, then there is no need to go in these breeding track of that animal or a wild location to collect the samples, because the getting of samples from wild animals is really difficult. So in that situation, if we have the somatic cell biobank, then in future there are several applications, and now there's the CRISPR also is coming up. So you don't know in future what will be the application. So anyway, we attempted this the project I submitted when I was at CIRB to establish the somatic cell biobank, the elite buffaloes. So in buffalo, we have the breeding program in the country. So we collect the somatic sample elite in the terms of those animals that are giving the milk more than the 3,000 liters, and the bulls. These are the parentage verified as well as from the progeny tested. So we establish, as per as the international guideline, at least 15 animals to be the cryopreserved in the biobank. I think Dr. Sambashiva is much aware of that. So in this project, we cryopreserve at present 52, but we are continuing this cryopreservation process of the elite animals, particularly for the breeding bulls of these Murra and Nili Ravi. We have some of these examples from the Assamese buffalo, because that was a project for us. And at least we have cryopreserved 25 vials from each animal, and these all the cataloging, which cell type, which date, what passage, what are their microsatellite data, so that in future if there is a cross contamination happens, because what happens in laboratory, there are several students, several people that are working. Possibility is that there is cross contamination of the cells. If the cross contamination happens of the cells or mislabeling happens, what happens when you use this donor cell for the cloning? Because in cloning, one somatic cell is only used, and if this happens, then there is a possibility of the mismatch which we are expecting. that clone to be born of that animal, but that was not, because that could be happen to the cross contamination of the somatic cells. So to avoid that, what we did, we immediately, when we collect the sample on the same day, we collect the blood sample from the same animal, and immediately that came to the microsatellite data generation. So in future is that if that animal we clone from our biobank, so we can match all these animals, which animals cells has been used to produce these clone animals, if there is any mismatching happen. So that is very important aspect we need to consider while making the biobank. So at present, from this biobank, the 5 animals were used to produce the clone embryos, and that also proves that, for example, at present in DRI, we have 4 pregnancies to be delivered in December. In that, these are all the cells from the biobank from the SR. So they are frozen cells. So we brought it here at NDRI. We thawed them. Thawing and the culturing them is simple. There is not much effect on the embryonic development. So this also proves that the cryopreserved cells work well for the production of the clone embryos. So I conclude that my talk is that we demonstrated the interspecies and the interspecies interbreed SCNT using the buffalo site, and we hope that the buffalo cloning is a viable tool or option for conservation of endangered bovines. And I'm really looking eagerly for these. If there are any samples, we get an opportunity to explore the possibility of the cloning of endangered, particularly wild animals or endangered bovines too, so that can be done. This is a talk from my talk. So I would like to acknowledge, because these are all the assisted reproduction technologies, so we require the generous funding. Without that, it's not possible. So my majority of funding, our laboratory is sponsored by the ICAR, the Indian Council of Agricultural Research. Some of the projects are funded by Department of Biotechnology and DSP, but these are majority. None of the projects is funded for the conservation of the endangered bovines. These all are funded for the domestic animals. So we extrapolate that information to the wild animal conservation. Also, one project is funded by the government of Chhattisgarh and Wildlife Trust of India for the wild buffalo cloning. So, and I thank you to Gopal Das and his team. He's a professor at the Animal Genetics Breeding at Guwahati Veterinary College, for his generous support to collect the tissue biopsy samples from the Assamese buffalo. Without that, we could not able to produce the Assamese clone bull at our place. Also, other collaborators, Dr. Kaluri, who has been associated with us for the horse cloning attempt, and my team, other members. This is not an extremely work of my alone. There are a lot of people they are working. So our team members from my previous institution, CIRB, as well as my current institution, NDRI. Thank you all. Thank you very much. I would like to take some questions. I hope I will be able to finish on the time.
Yes.
Or I will finish early.
Yeah, quite early, I would say. Yeah, a minute and so. But thanks a lot for giving time. So a quick wild thought from me. So this Assamese buffalo, what you have shown, how we have cloned in the field, and this technology, really nice and enriched talk, I would say, with respect to cloning technology. So I was wondering, like yesterday we had a talk by Joseph Saragasti. So he was talking about not white rhinos. So is it possible? I mean, there are only 2 individuals left, and then is it possible to have another host closest, let's say southern right rhino, and use the same cloning technology and try to fix the species? I mean, just a wild thought. Your comments.
Yeah. But you see, so if there are rhinos, if somebody wants to clone, it has been the possibilities that we can explore it. We are not sure that embryos will develop to the blastocyst stage or not. I think if you see my talk, so we miserably fail in horses. Okay, we could not able to produce an embryo. Possibilities that in rhinos also no embryos will produce. But if the embryos produce, then the second possibility is that we need the recipient animal. That's also an important aspect, because the embryos, even if the embryos produce, for example, I see a lot of papers, it shows that the cattle oocyte is the best reprogramming ability as compared to any other domestic oocyte. So people, they produce the cattle clone embryos using human cells also, or from other wild animals. Yeah, the people, they produce, and they produce the stem cells from these clone embryos to produce That's what I was saying, is that it's really important, because nowadays people are talking about the production of the sperms and oocytes from the stem cells. Maybe after 30 years it's possible. So it's a good idea to explore, at least to produce and clone embryos from the rhinos. If in India they have, so we need just a somatic cell to produce and try to generate at least in stem cells and make them in biobank. I think CCMB is doing excellent work on the biobanking, as I know. So then make them in biobank in future, when we have the technology of the generation of the oocytes and sperms from the stem cells, then there will be excellent opportunity for the conservation. This is my view.
Thanks. Sounds great. So we have a couple of questions. So could you elaborate more on how clones have been produced using stem cells, asked by Nicole Tay. So he just, wants to know, I think, more detail about how you can clone using stem cells.
Yeah.
So
For the stem cell, I think this is similar to the somatic cell. Only we need to establish the stem cell. So for example, the embryonic stem cell to be isolated from the inner cell mass of the blastocyst. So cut the blastocyst inner cell mass and seed it on the mitomycin treated feeder layer, and then treat it with the growth factor FGF allied the growth factor required when the cells they expand. For example, the bovine stem cell, they form the colony and the dome shape. And after that, you trypsinize this cell to make a single cell, and the single cell to be used as a nuclear donor to produce the clone embryos. The only difference between the stem cell best cloning as a somatic cell is that your cell source is different. The rest is the method is similar. Absolutely, there is no change.
Thanks. Next question: why bovine species have better success rate for cloning? By Nishta Bhati. Sorry. So
Yeah. If you see, because the majority of the people they work on the bovine in domestic animals in all these countries, European countries, USA, Australia, they work on the bovine. So that's why the more refinement has been made in the bovine. There are a lot of nowadays. If we talk, I think India also talking about the IVF test tube baby in bovine. There are 30 institutions that are working on these IVF centers in Andhra. So I know there are 2 centers that are working on the IVF funded by government of India. So OPU IVF, obviously OPU IVF. So why it is successful? Because there are a lot of people that are working in this bovine. There are defined formulation of media, defined protocols. If somebody works, lot of people working in buffalo, it works well. Absolutely is not possible. Also is that in bovine, there are the literature information is available, or you get a large amount of sample for your experiment. Other species, there is a little bit difficult to get the biological samples for experiment. This is, I believe that's why the bovine is more successful.
Thanks. Next question is by Raj Mohan. Can you please explain behavior changes and offspring anatomy with respect to gene expression, protein expression, and maternal inheritance in clone program of domestic cattle oocyte with wild somatic cell nucleus?
Yeah. This is, I think, a good question. I appreciate it because we also find that, for example, we have not much data on that, but I would like to share, for example, our Assamese buffalo. This in Assam is this bull is available in free ranging, so it's very difficult to handle. But in our situation, when we produce the clone using domestic animal oocyte of that and clone, and then here these animals still they are aggressive, but not that aggressive. So it is might be an interesting question for us to examine the expression pattern of imprinted gene or epigenetic. And obviously, these are mitochondria. These all may affect the behavior of that animal. So we don't did any studies on that, or also nobody has did these intensive studies on particularly in the wild animal using this approach. But this is an excellent idea to explore in future that what will be the effect of this imprinted gene mitochondria on the behavior and the performance of these animals to be cloned using the domestic animal oocyte. And moreover, very few studies have been done for the cloning of the wild animals using the domestic animals. That is also a limitation. But this is a good option to explore in future.
Thanks. Dr. Jimopati has raised hand, so please unmute and you can ask.
Thank you. Wonderful talk, Naresh, and a lot of work you people are doing. I just want to know, did you examine mitochondrial genetic relationship with reference to the wild versus domestic? When you clone, all the mitochondrial genome comes from domestic animal. So have you before tried to understand how they are different from this both mitochondrial genome?
Yeah. So this is recently we attempted it. We did the next generation sequencing. So this we particularly not did in wild animals, but in this study we include the buffaloes clones as well as Assamese buffalo we also included. So to see from exactly where these mitochondria are coming or what is the mitochondrial inheritance pattern. And this was a question in many platforms to people they ask, and it's absolutely perfect questions, I say. I think maybe in the 6 months onwards, we get in published it. We analyzing the data, we generated the next sequencing data. We have the data of these 10 clone animals because 7 clones from a single animal. And this is really important to see that each animal, they usually, if you see in embryo production in our method, we use the 2 enucleated oocytes, 1 somatic cell. So possibility is that we can get the mitochondria from 3 different sources. This is one possibility. Might be from 2 and might be from 1. If we get, and what was the percentage? So we are doing it, and I think 6 months we can get an exactly idea how much, but that was already done. So we have the data, the raw data, we are analyzing it.
Yeah, that will solve the many questions people are asking about behavioral epigenetic modification, all kind of behavior. Yeah, thank you so much.
Yeah.
Thanks. There's another question by Raj Mohan. So what is the reproductive changes of next generation offspring from clone animal? Is there any crossbreeding studies of clone animal with wild or domestic one? Any study on genetic pollution in the wild?
You see, normally, as Dr. Umapati said, first, if you have clear that what is the mitochondria is there, if it's clear that mitochondria from the wild part, then it's absolutely, there is no pollution in wild animals because we enucleated the oocyte from domestic animals. So only question is that we have to make sure that the origin of mitochondria, which is the genetic pollution, we say the genetic pollution in wild animals. If you make sure is that there is no genetic material coming from any domestic animal counterpart, I think mitochondria, because the source of genetic is only mitochondria. Rest we have enucleated it. So it will be no effect on these genetic pollution. Other aspects, for example, the fertility and other thing, as we mentioned, is that, for example, we have Assamese buffalo here, and this bull is producing semen, but this bull is producing semen, producing embryos to IVF, and now we are planning to use this bull semen for the AI or for the natural AI, particularly AI, because this bull cannot go to the Assam and the northeastern part. So if you did the breeding well there, then the fertility is absolutely not a problem. Because once the bulls they mature well and producing semen, it seems that this bull is absolutely normal. What happens usually in cloning, if there is any abnormalities, I accept is that in clone animals they have abnormalities. There is absolutely is a well-established fact. We also notice that in our buffaloes. But when the animals they are mature well, producing semen or producing milk, they are absolutely fine. There is no absolute abnormality. Even though if they produce the progeny from that animal, this animal progeny is absolutely normal. We have produced around the 50-60 progeny from the clone bull semen, and we have the 4 clone females at NDRI, and they are delivered the calves, and their progeny is also giving the milk. They are absolutely normal. So our data or our experience says that the progenies of this clone animal, those are matured well. I'm saying there's a question again, is the maturation is important. If they matured well, producing semen, then they're absolutely normal.
Thanks. There's another question from Nicole Tay, probably last for the session. Have you found data efficiency in blastocyst formation using ESCs in nuclear transfer?
Yeah. Yes. We did. This is for the in buffalo we did that. In wild animal we don't did, but ESCs is far better with program as compared to the somatic cells that we did in buffalo. So embryonic blast reproduction rate or reprogramming ability of the embryonic CSC, the embryonic stem cells, they are far better as compared to the somatic cells.
Thanks a lot. I think we don't have any more questions. So I really thanks Dr. Naresh for a wonderful talk. And so a couple of announcements before ending the session. We have an excellent panel discussion in the evening around 4 o'clock. So be there, be available. And we'll be posting a link for feedback form. So all participants should write the feedback form, and at the end the certificates would be given on the basis of that. So please be available for the panel discussion at 4 p.m. Indian Standard Time. Thanks a lot, Dr. Naresh.
Thank you very much, and my personal thanks to Dr. Umapati and Dr. Sambatu, and good luck for the team at LaCoons, and everyone at LaCoons, my good wishes. Thank you.
Thank you, Naresh. Thank you.
Thank you, Naresh.
Thank you. Okay, sir. Thank you. Bye.
Yeah.